Welcome everyone to today's edition of the EV Journal Club, and what a special edition it is. Today the Journal Club is being co-hosted by the Student Network on EVs, or SNEV, and I'm actually so excited that I'm wearing a tie today for maybe the second time. I mean, you have to look beneath my beard here, but maybe for the second time in the last 6 months. So just a few words about SNEV. This Student Network on EVs was just formed this year. It had a first public meeting at the ISEB 2020 virtual event. And this is really a way for students to be able to connect with each other, to find opportunities. The network's president is Amir Nasiri Kanari, and he leads a large board of students from all over the world, from Australia, where he's located, to Iran, and really around the globe. So you can learn more about the network by visiting snevresearch.wordpress.com. And I will put that in the chat box too for those of you who didn't catch it. On this journal club, as with some of our other journal clubs, we have the privilege of welcoming some of the co-authors of the paper that's going to be discussed. Alyssa Weaver, whose lab produced this paper. Alyssa is here. She's Alyssa, if you want to just say a couple words and maybe introduce Bonghwan as well.
Sure. Hi, everybody. My lab studies extracellular vesicles, mostly in cancer, and we have a big focus on migration. And Bonghwan Sung, who's going to introduce himself too, really has driven most of the migration work in my laboratory and did virtually all of the work in this paper.
I'm Bonghwan, work for Alyssa Weaver's lab, and first author of that paper you will discuss. I'm really happy for you guys to discuss it. And I'm working for developing reporter to track exosome secretion and uptake, and also working on the cell migration stuff regulated by EV secretion.
Excellent. Well, Bonghwan, thank you for joining us, and thank you, Alyssa, and we're looking forward to the presentation and then some discussion too of the paper. So it's my pleasure now to introduce my SNEV co-moderator for this session, Will Hotham. So Will is coming to us from Cambridge University in the UK, and Will, I'm going to hand this over to you so that you can introduce our presenter.
Brilliant. Thank you very much, Ken. Thank you for the introduction for SNEV as well. So today we've got Ashley speaking to us. She's a third-year PhD student in Dr. Godwin's lab in the Department of Pathology and Laboratory Medicine at Kansas University. Ashley got her MS in Microbiology, Immunology and Molecular Genetics at the University of California, and she's currently researching a project looking for biomarker potential of non-coding RNA encapsulated by extracellular vesicles, specifically in the context of cancer. However, she's also got quite a broad knowledge of various other aspects, including soybean genetics, algae cytoskeleton, mTOR, and Toxoplasma gondii. So quite a good breadth of knowledge from Ashley. And she's going to be presenting this really exciting paper. So I think over to you, Ashley.
Hello, everyone. I'm glad you're choosing to be here today to go over this pretty awesome paper that I think will be of a great resource for extracellular vesicle scientists and researchers. And it has a lot of, I think, interesting potential to better understand the dynamics of cell-cell communication in the extracellular space. Hopefully everyone can hear me okay. Just let me know if I'm using a weird owl that responds to my voice, so hopefully it should be able to pick up everything. So today we're going to be discussing a paper called A Live Cell Reporter of Exosome Secretion and Uptake Reveals Pathfinding Behavior of Migrating Cells. And I'm Ashley Tetlow. I'm, like as he said, in Dr. Godwin's lab at University of Kansas Medical Center here in Kansas City, Kansas. So let me just move my little thumbnail here. As you probably all know, exosomes are very important membrane-bound vesicles that are traveling through the extracellular spaces between cells. They're involved in a lot of different processes, including cell-cell communication, as well as migration, and they are typically generated through this endocytic pathway, in which late endosomes become vesicular bodies through the invagination of the intraluminal vesicle into these exosome-sized molecules that are then secreted into the extracellular space and become what's known as exosomes. And they are absolutely jam-packed with different biomolecules that are important for cell signaling, as well as representative of their endocytic origin, such as tetraspanins. One that we'll be focusing on today in this paper is CD63, a 4-pass membrane tetraspanin that is found on a large percentage of exosomes. And they also contain a number of other biomolecules that I'm sure you guys have heard a lot about, some of which are ALEX and TSG101 that are also kind of covered in this paper a little bit, and then HSP70. So exosomes are of endocytic origin. So what they did in this paper is they were able to adapt a tool that was used in synapses to look at action potential. So basically taking these pH-reactive GFP molecules and looking at actual action potential firing through the endocytosis of these vesicles. And then they use this GFP molecule called ecliptic fluorine that is called ecliptic because it, when the pH is lower, it stops fluorescing until it is the pH is reestablished to a neutral state, and then it begins fluorescing again. So it's great for studying extracellular vesicles and also synaptic vesicles. So just to show a little bit about what this sensor does actually. So wild type GFP is pretty stable at pHs between 5.5 and 7.5. It fluoresces, emits wavelengths of light in a particular pattern. Whereas there's also ratiometric fluorine, which at different pHs will emit a different color light. And then there's also ecliptic fluorine, which I mentioned, quenches its fluorescence at the lower pHs. So a great tool to be able to study acidification of vesicles and also secretion and synaptic transmission. So move this back over here. Okay, so in this paper, they mainly look at 3 different types of reporters. Their original reporter, which was reported, I think, a couple years ago, called fluorine CD63. So that takes the CD63 tetraspanin, and they attached a GFP, the ecliptic GFP molecule, into the first extracellular loop here. This larger extracellular loop is known to be involved in the dynamics and functioning of CD63, so it was smart to choose this smaller loop here. And then this is just representing that the fluorescence is turned on and off in response to the pH of the GFP molecule. And then they also mentioned that there was a paper that in a bacterial fusion protein showed that this M153R mutation stabilizes the GFP molecule in vivo. So they were able to do site-directed immunogenesis and were able to add that mutation, and also showed increased stabilization in vivo. And then they also have this third dual reporter, which is, I think, the most interesting, which it's probably incorrectly demonstrated here, but it's just got an RFP, red fluorescent protein, M scarlet, and GFP, which responds to the pH, making it an interesting and dynamic dual reporter for exosome secretion and multivesicular body trafficking. CD63 is a pretty good exosome marker as far as exosome markers go. There's a lot of heterogeneity in tetraspanin representation in extracellular vesicles, but as we saw recently in David Leiden's paper a little while ago, he showed that through different mass spec of these different exosome subpopulations, exomeres, exosomes small and exosomes large, they were able to show that CD63 is pretty well represented in small exosome fractions, as well as CD9. It's a little bit less well represented in some plasma, human plasma, but overall it's a pretty good exosome marker, and this is just showing the overall structure of CD63 and how it sits in the membrane. So starting off with figure 1, we wanted to take a look at the acidification of multivesicular bodies as they bind to and push their contents out into the extracellular space. So they labeled CD63. This is just CD63 with the gray boxes representing the extracellular loops. The gray box with the underline being the first extracellular loop. So they inserted the GFP moiety here. And then the R represents that mutation that I mentioned a little bit that was used in a bacterial fusion paper a few years previously that showed that it stabilized the reporter. And then this is just the diagram showing how it works. It's doesn't fluoresce in the acidic space, and then once the fluorophore is exposed to neutral pH, it starts fluorescing. So figure C is just showing their two exosome preps, or sorry, large extracellular vesicle and small extracellular vesicle. So this represents the 100,000 times G pellet, which usually pellets small extracellular vesicles, with the majority of them being extracellular or exosomes. And then large EVs, which can also include microvesicles and other large vesicles like apoptotic bodies sometimes and things like that. So this is showing their quantity and size. And then they went ahead and did some Western blots showing that you can see the glycosylated CD63 in the parental as well as the fluorine construct. So these are HT1080 fibrosarcoma cells that are often used in cell migration studies. So this is just showing total cell lysate, the representative bands of CD63. And then they showed in the large exosome prep that there's hardly any that I can see CD63 represented. And then they showed in the small extracellular vesicle fraction that there is not only CD63 present, but it's also shown at this higher molecular weight, demonstrating the shift of 27 kilodaltons because of the addition of the green fluorescent protein. So then showing kind of what's typical of exosome papers is they usually have to show several markers that reveal the origin of the exosome. So TSG 101 is a canonical exosome marker that's used a lot. So you can see that it's represented in the total cell lysate as well as in the small extracellular vesicle fraction. GM130 is a cis-Golgi stack protein that should not be put into extracellular vesicles. So you see that it's only found in the total cell lysates from the parental and the fluorine-associated HT1080 cells. And then you have flotilin as well, which is also sometimes expressed in microvesicles, as you can see kind of here, but it's also represented in exosomes. So now to get to some imaging of the reporter itself, this is just showing the reporter in live cells as they deposit extracellular vesicles along the coated dish. And then mCherry, this is just labeling the plasma membrane to show that the vesicles that are being deposited onto the dish surface are actually of this CD63 positive population, and they are not actually plasma membrane just being deposited behind the cell. And this is a kind of still image of the old reporter, the reporter without the site mutation, showing, you can see the basic activity of it here in kind of like a amorphous blob, but it's hard to actually define punctate regions So I think this reporter does a lot better job showing up on these kind of live cell images. And this is just a little movie of that still image there from the previous slide. And it's really cool to just observe how the cell is able to move across the space, and then these retraction fibers leave extracellular vesicle punctae behind it that generally do not move after being deposited onto the dish here. And you can kind of see it in this older reporter, but it's also a lot dimmer, and the punctae don't show up as well as they do in the new reporter. So then they had to show that this reporter is labeling exosomes. So once again, they just show the size distribution of their exosome isolations, large and small. And then they did anti-GFP immunogold labeling on TEM negative stain. And this is something we've also seen in our lab with immunogold labeling. Tends to reveal a little bit of the heterogeneity of the exosome population. So you can see that not all of them are CD63 positive with the GFP, and not all of them are able to be labeled with the gold particles, but a good percentage of them are, and they are demonstrating the correct size. And then they did an ion-dixonol density gradient in order to further define these EVs. So since we know most EV fractions are of heterogeneous population, fractionating can give you a good idea about if proteins are actually co-localizing to the same vesicle, or if they're actually being associated with other things like small apoptotic bodies or exomeres or something like that. So this gives us pretty good evidence that CD63 is in this fraction 6 from this density gradient, as well as flotilin. And then you can also see GFP showing up, as well as HSP70, which is another exosome marker on the inside. And then GM130, the Golgi marker that should not be contained within extracellular vesicles, is not present. So then, in order to show that these are actually exosomes, they went ahead and did a shRNA knockdown on the RAB27A gene, which has been previously shown to reduce exosome number and but not change the size or morphology or protein content of the exosomes, just reduce the number. So they performed a knockdown, a pretty efficient one, as a matter of fact. So you can see that there's hardly any RAB27A being detected here. And then they're showing the number of EVs per EV number per cell per hour over time. So this is just showing that the numbers of EVs produced by these knockdown cells is significantly reduced. And then this is just essentially taking a section next to the cell, thresholding the area, and then examining not only the threshold area of EV secretion, but the intensity, to show that the area of EV secretion is reduced significantly, as well as the actual intensity of the EV punctae is also reduced. So in F, you can see the scramble. That's a little bit hard to see, but you can see EVs lighting up on the surface of the dish here next to the cell that's been deleted from the image. And then you can also see in the knockdown that there's basically little to none of that effect. So you can pretty well conclude that RAB27A knockdown reduces exosome production and is reducing these CD63 positive vesicle that have been shown to be attached to the plate here. Okay, so now this is just kind of like further evidence showing that these are marking secreted vesicles. So they're doing some co-localization experiments. And just to note, the PFA fixation causes the vesicles inside the cell to also fluoresce just because of permeabilization. But in this box here, they're showing their reporter co-localizing about 20% of the time with TSG 101. So these arrowheads are pointing to double positive CD63 TSG 101 vesicles. So we know that exosome markers are pretty heterogeneous in their representation in every vesicle. So this is pretty well in line with what we've seen before as far as exosome marker co-localization. So you can see about 20% of TSG 101 punctae are also CD63 positive. And you can see a higher percentage of ALEX and CD63 positive punctae as well. And then this is also just showing the ALEX co-localization is at a greater extent than the TSG 101. And then in part C, they're focusing on these retraction fibers at the rear of the cell. So this square eye here is enlarged in this frame. You can see that there's this kind of trail of extracellular vesicles stuck to the dish here of various size, this being quite a large one. That we see down here is not fluorescent with this CD63 marker, so it's probably a vesicle of some other origin, maybe a microsome or a microvesicle. It's difficult to know. So then they're also showing that these vesicles that are lighting up along the retraction fiber tail edge of the cell are of the appropriate size to be considered exosomes. So that's pretty good evidence that these are CD63 positive exosomes that are being deposited onto the surface. And then these larger vesicles are not being labeled by this marker. Therefore, they are likely originating from some other type of extracellular vesicle creation event. All right, so figure 4, I find this part really interesting just from like a biology perspective. They were able to, I know that a couple of papers before this, even I think one by Alice Weaver's group showed this interesting migration behavior that cells exhibit along exosome tracks. So this is just showing their plasma membrane marker on the panel at the top here, and then their exosome reporter, and then the merged image. So it's a little bit easier to see in the video, which I'll show next, but these protrusions from the cell's leading edge are over pockets of exosomes, and then it sort of uptakes some of them and decides which direction to go based on these exosome trails that it's exhibiting leader and follow behavior. So what they did is they measured the angle, or they did a cosine of the angle of the apex of the cell towards the exosome track, and were able to show that a pathfinding index of 1, meaning there's 0 deviation from the cell on the exosome trail. So you can see that the majority of the cells are migrating towards, on top of, and over exosomes left by other cells. So that's really interesting. They're not, there's very few, maybe a couple cells that are exhibiting migration in the opposite direction. So this is on a 2D surface. So interesting behavior in a 2D environment, but they were also able to show that this happens in a 3D environment, which is interesting and important for cellular behavior in vivo, being able to show that, yes, these cells do migrate toward and over these exosome deposits on the surface here. And then they were able to show, so this is some mouse work using breast cancer cell line MB251 injected into the 4th mammary fat pad. So they used their reporter positive breast cancer cells, let a tumor grow, and then did some imaging of these cells and of these exosomes in this environment. And you can see that there's these collagen fibers in the extracellular space that tend to act as the substrate for exosomes to kind of stick to. And then there's also cells will crawl over and across these exosome deposits in a similar pathfinding behavior to the 2D dish. And you can see panel I here showing the exosome deposits at the end of the cell, and then these are showing retraction fibers, adhesive protrusions also. And then another in vivo experiment kind of demonstrating the reporter utility of this in vivo is using chick eggs or chick embryos, injecting their reporter positive exosomes into the chick vein, and then taking the plasma and looking for their reporter positive exosomes over a period of time. So this is an important piece of exosome research, I think, is looking at how long it takes for these exosomes to either find a target cell or basically be reuptaken. And it's actually surprisingly a very quick process. Even in this chick embryo, they're showing that there's a half-life of about 15 minutes for these exosomes in a in vivo chick system. So it's their signaling is very rapid, it seems, and that might be important for implications in biomarker studies that tend to rely on circulating extracellular vesicles to reveal disease states. So a stable reporter that can be identified in the live animal over time is very useful, I think. And so this is just kind of showing the cells. So this is a 2D, I believe, yeah, 2D environment where the cells are able to crawl over these exosome deposits left by other cells. And this cell is really interesting here. It sort of comes this way, has almost 3 choices it can make as far as which exosome trail to go across, and then sort of investigates, uptakes some of them, and then decides to go towards the most concentrated, it seems, amount of exosomes. So I don't know exactly what's going on in the cell that's behind this behavior, but I just find it very interesting. Like, can you do co-cultures with other cells to see which exosome trail is most attractive to a migrating cell, or is this just kind of some sort of generic event where it's utilizing the adhesins that are deposited onto these dishes by the exosomes to move itself along? So it's a very interesting cell behavior, and you can kind of see it's the leading edge ruffling along these exosomes. So that's in the 2D environment. It's a little bit easier to see in the 2D environment, but the 3D is also very intriguing. It also has sort of this decision-making moment where it's moving through the space and decides to go this way, and you can see another cell coming along. So those are just great. I love those. And then this is a 3D reconstruction of the mouse and mammary tissue exosomes. So you can see, it's a little bit hard to see, but there's these punctae and this Z-stack image of the mammary fat pad. So showing exosome deposits in actual live tissue. Well, in this case, it's not live anymore, but in living tissue. So figure 5 is showing that basically the exosomes are deposited at the front edge of the migrating cells on a nanopattern dish. So they said they couldn't rule out exactly if exosomes are being deposited on the rear edge of the cell as well, but it is fairly clear that they are being deposited on the front edge of these cells. So this is just kind of a 3D patterned dish, so it has like nano ridges in it to create a little bit more of a rigid pattern for these EVs to fall into, and it's a little bit easier to study their dynamics on a dish like this, on a nano pattern dish. So they looked at a cell migrate across this dish with the reporter present, and were able to do time lapse of the exosome deposits, and we're able to show that the exosomes are being deposited at the front edge of the cell, and they tend to stick, and there's very little movement or reorganization after they're deposited. And the video shows it a little bit better, but you can see as the cell tracks through here, they tend to not move very much after they're deposited, and they're deposited in these like linear grooves. And then they also show the displacement length is quite small, so there's tends to be the majority of them are not being displaced after deposition. So this is just the video of that figure. So you can see as the cell moves across this space, it's depositing exosomes in this nano pattern. So another addition to this great tool is that they added a mScarlet tag onto the reporter here. So they're kind of just showing that the mScarlet tag doesn't interfere with the function or the localization of CD63 in vivo. So here they have their reporter, LAMP1, which is a late endosome or lysosome marker. So they're just showing intracellularly that the LAMP1 is co-localizing with CD63, which is what it is supposed to do in a normal typical cell. And then they're showing their dual reporter also co-localizes with LAMP1. So I would just focus on the zoomed in images to see the co-localization. And they also quantified it for you here. So percentage of LAMP1 co-localization with the single tag fluorine, the dual, and mScarlet and the fluorine, a part of the dual tag. So there was a significant difference between the single tag and the double tag. They mentioned that it could be something about degradation of the single fluorescent channel. And then Rab7 co-localization. Rab7 is also important in the endocytic pathway. This is just the images that go along with the Rab7 graph from the previous slide. So they're showing CD63 co-localizes with Rab27, or sorry, Rab7 in the endocytic pathway in the same fashion that it does in wild type or parental cells. It's fairly consistently co-localizing with the proper proteins that it's supposed to co-localize with. And then they were able to also look at the extracellular deposition of these reporter positive exosomes. And then they were showing that it is the reporter and the secondary reporter, the mScarlet and the CD63 are all co-localizing between 80 and 100% of the time. So there doesn't seem to be any defect for adding an additional fluorophore here, which is great for studying in vivo effects. They did mention that there was a slight upregulation or increase in stability of this reporter that could, maybe affect some biological processes, but they also offered a couple suggestions for how to combat that, maybe by doing a site-directed mutagenesis of endogenous CD63 to allow for the stabilizing mutation. And then, so in this figure, they're just looking at the dual reporter multivesicular body transport before and after endosome uptake. So this, I think, is very interesting to look at here. We have a 60-minute video and a chymograph showing some of these events. The pink arrow showing multivesicular bodies after uptake, so that they're in the process of being uptaken and reacidified. And then the white arrowheads show filipodia contact with the extracellular vesicle or exosome deposits. And so these are just some images from the chymograph over time. It's very, very interesting to see how long it takes and what are the dynamics looking at these events here. So an exosome will be deposited by perhaps another cell onto the dish, and then another cell comes along. Its filipodia tend to search out for adhesions and places to sort of attach to. It finds an exosome, and then it's typically, once it touches the filipodia, is rapidly uptaken and reacidified. So from contact to uptake, it takes only about 6 minutes on average for an extrasome deposit to be uptaken by a cell through its filipodia extensions. And then this is just a still image of a video that I'll show you in a second, just showing these punctae being exosome deposits being uptaken into a cell and then being acidified over time. And then you can also kind of see perhaps a engulfment of a vesicle at the top here with this top arrow. So I'll just play the video here. You can see that even inside the cell, there's a lot of vesicular trafficking towards the cortex of the cell, and then also outwards. And then as the cell crawls along this space, it's uptaking these stationary exosome deposits. That one was just uptaken. White meaning that it is neutral pH, pink meaning it's acidified, and you can see it rapidly turns pink and disappears inside the cortex of the cell. And I think this behavior is quite interesting as to like the fate of these exosomes. Is the cell opening these up, examining the biomolecules present in the exosome somehow and making behavioral decisions based on these exosomes, or is it really just a anchoring event, or is there some kind of processing to where a cell decides to take up an exosome or not? It seems to be here that it just takes up whatever its filipodia encounters. You can see there that the cell membrane extension there just touched the vesicle, and now it's rapidly uptaken. So this process is very fast, which is interesting and surprising that with under 10 minutes these cells are uptaking and processing biomolecules from the extracellular environment and making decisions about it. So as someone who studies exosomes as biomarkers, part of me is wondering how exactly the biomolecules encapsulated within these exosomes affect a cell that might come along later. So how would a cancer cell's exosomes affect the behavior of a neighboring cell? Or a lot of exosome research involves using conditioned media from cell lines, or even 3D culture. And then how many of these exosomes are we missing that are just stuck to other cells, stuck to the dish? is there a certain dynamic of which exosomes are more motile in the extracellular space, or is there a certain population that's mainly used for this like adhesive migration behavior? It's all very interesting, and I think this dual reporter could reveal a lot in the future about how exosomes are being trafficked in the endocytic pathway inside the cell and outside the cell, and how those influence behavior. And I think there's so many experiments I've come up with just after reading this paper, just out of my own curiosity to look at how these cells are using exosomes to do cell-cell communication, and what is the fate of an exosome that's secreted by a cell, and what's the fate of its biomolecule in the end. So yeah, it's a very interesting paper, and I think it'll be a very useful tool for a lot of exosome biologists to look at live cell imaging. They were able to do intravital imaging in the mouse mammary fat pad as well as the chick embryo. So very, very well established in vivo reporter, I think, as well as a in vitro reporter. You can think of a lot of different experiments and how to use this to look at not only exosomes, but how exosomes change cell behavior and cell migration and processing and everything like that. So with that, I'll say thank you, everyone. And I can pull up any slide if you want to specifically talk about it. And if you have any specific questions, I think that, as was mentioned at the beginning, the lab director who published this paper and the scientists who did most of the work are also on this Zoom, so they would have very specific answers for you, I think. So thank you.
That was really, really nice, Ashley. Really well presented. That was fantastic. If people want to send their questions into the chat box, I'll go through them, and then if you want to sort of unmute yourself and ask your question either to Ashley or to the lab members. So the first one was from Anne. Anne, do you want to unmute yourself? Anne Wayman, regarding the
Thanks, Will.
clem data.
Thanks, Ashley, for a really nice presentation. So I was looking at the clem data, I can't remember, figure 2 or figure 3, somewhere around there. And they were showing with this really just beautiful technique. So you see these retraction fibers that are starting to break up a little bit. Yeah, exactly, this figure. And then you see these strings of exosomes. And I'm wondering how they decide when it's a retraction fiber when they're looking at just the fluorescence data without the electron microscopy, and how they decide that it's just a couple exosomes sitting next to each other where the fluorescence is merging together. So this is really tricky data to analyze without the electron microscopy, and I was wondering if they had some tricks they could share with the rest of us.
I mean, I guess I can make a couple comments and Bangwan could add in. So, I think there was a movie, Ashley, that you showed that went along with figure 1, where you can see the cells sort of actively migrating. Maybe the top one, right? And what you can see is that the cells are sort of sticking to the puncta and have these retraction fibers, and they're reorganizing those puncta as they're traveling over them. So, so I think, it really requires live imaging, and it requires, good enough high resolution microscopy to really be able to discern whether something is a thin retraction fiber or a puncta. But you're right, it's challenging.
And then is there any idea why CD63 is in the retraction fibers? Is it just in the plasma membrane generally?
So that's been my thought. Do you have any additional thoughts on that, Bangwan? Or?
Well, my thought, another my thought is that deposition of the exosomes after secretion at the leading edge can be reorganized or realigned at the retraction fibers at the tail of migrating cells. But that's just my presumption, and we don't have kind of very clear evidence why the CD63 is associated into retraction fiber.
Thanks.
Thank you. And that sort of leads on to the next question from Natalia. How do the authors know these are exosome tracks and they look very similar to microsomes?
Yeah, they mentioned in the supplemental that sometimes there are these deposits of they look like packets of exosomes. So the membrane surrounding them, I believe, is labeled with the CD63 reporter, whereas the vesicles inside are dark. So it's been shown, I think, in like a 2012 or maybe 2011 paper that microsomes are associated with the retraction or adhesions of cells. So they're just basically like sacks of exosome-like particles that are left behind as the cell moves along, sort of stuck to the surface. But they are to be CD63 negative. So I think they did mention that there is evidence of microsomes in some of these cells moving across here. But it was in the supplemental materials. And I don't think it's observed very often, correct me if I'm wrong, but I think the microsomes tend to be fairly rare, and the majority of what we see, at least in this paper from what I could tell, is the exosome deposits.
Brilliant. Does that answer your question, Natalia?
Yes, my question was related to the previous one because the microsomes are released from the retraction fibers, so and it looks pretty similar to the pictures that I saw in a previous paper. So that was my question. I wasn't sure if they look like microsomes or exosomes, but okay. It's fine.
Yeah, so microsomes are like big, right? They're about the size of multivesicular bodies, whereas we saw it once in a while, but mostly, Ashley's right. She read our paper really well. Mostly we see little exosome-sized vesicles.
Perfect.
And there was one of those recent microsome papers from the past couple of years. They knocked out or knocked down a whole bunch of different tetraspanins, and CD63 did not affect microsome formation. So we think that microsomes are tetraspanin related, but that they're different tetraspanins from CD63.
Perfect. Beautiful work. Congratulations. I love it.
Thank you. And Fabio, your question, if you want to unmute yourself.
Yes, I just was wondering if you studied the composition of the EVs that the cell decide to follow, and if you see an autocrine or paracrine absorption or uptake of the EVs.
So can I answer?
Sure.
So actually, that's what we are doing right now. We didn't we didn't do investigation into the cargos, the promoting the directional migration. 2017, we published another paper in Cell Adhesion and Communication showing the chemotactic activity of the exosomes or small EVs to promote the directional migration of cells. We can presume, there are some cargos to attract directional migration of cells, but we didn't look at it. But obviously, the exosomes or small EVs carries many cargos in terms of receptors growth factors, any other stuffs promoting the cell migration. And about autocrine migration, you probably see the movie or the quantitation of cell migration on nanoparent dishes. So we can see, the CD63 labeled exosomes deposit at the leading edge of migrating cells, and the cell can use them to promote its migration. Also, another evidence of autocrine migration of that, if you genetically inhibit the exosome secretion by LAP27A or other ascorbyl protein knockdown, then you can see reduced cell migration. So that's how exosomes works in autocrine manner for cell migration.
Thank you so much. Nice work.
Thanks.
We've now got. Oh,
I'm sorry.
No, you go, Ashley.
I was just wondering if have you seen any evidence of a cell migrating over its own exosomes over, like, a period of time? Or is it usually like a leader cell and then other cells kind of follow?
Well, I think that's been hard to see, because as you can see from this movie in front of you, the cell pretty much migrates over the top of it and then leaves it behind. So I think the paracrine is easier to see when another cell left it behind. But I think we have a lot more work to do to really be able to distinguish when cells are migrating over their own tracks versus another cell's tracks that are adjacent. And it kind of depends on how long you take your movie for and when you're catching them migrate. So having this brighter reporter actually helps us a lot. Before, we were bleaching our cells so fast, we just couldn't take the movies to, or long enough movies and bright enough movies to really answer that question.
And we've now got a question from Kovashazi. If you can unmute yourself.
Hello, I am here, but I think I have made just some notes. I did not have questions, did I?
That's a comment. The composition of the exosomes. Yeah, I mean, I think it is hard to study. I think you'd have to combine techniques, right? Maybe have some other system where you have a specific cargo that you think is affecting migration, and then maybe combine a tagged version of that cargo with this reporter, something like that.
And we also have a question from Rad Jasmine, if you want to unmute yourself.
It looks like Rod may have dropped off, so why don't you just go ahead and that one.
So their question was, could we use this visualization technique to determine the in vivo lifespan of circulating cancer-derived exosomes? I don't know who wants to take that.
There are two things to be considered. So half-life of reporter or half-life or lifespan of exosomes its own. So this reporter is actually based on fluorescent proteins. So we can measure half-life of that fluorescent protein as shown in figure. But it doesn't mean we can measure lifespan of cancer-derived exosomes in vivo. It is really hard. I believe there's no method to measure lifespan of circulating exosomes yet. But that reporter can help how to track exosomes in vivo or circulating exosomes in vivo, but it may not decide or it may not measure the in vivo lifespan of cancer-derived exosomes.
Yeah, I mean, I guess we did the half-life of how long the exosome stayed in the chick blood, but that doesn't, we didn't know how long they persisted in tissues afterwards. And, you know, I mean, I don't know that our tool would be any better than a standard GFP CD63 for answering this question, and maybe worse than a luciferase. It's really designed as a live cell reporter for studying subcellular processes.
Yeah, I think it's pretty on par with what was been shown. It was an old paper, I think, looking at fetal-derived exosomes in maternal blood after giving birth. Their half-life is they're only remain in the mother for, I think, around 2 hours circulating. I mean, like you mentioned, it could be different in the individual tissues. I imagine it probably is due to the stickiness of exosomes. You know, as shown in this image here, they're attaching to collagen fibers, so they probably persist in tissues at some greater time frame. But at least in circulating EVs, it seems to be they're either uptaken or you know, degraded or something on the around 2 hours post introduction into the circulatory system. So it seems to be pretty short half-life in the circulation.
Does anyone else have any questions at all? I've got one very, very quick and very basic question, and it might have been covered in your older publication, but why did you choose CD63 to target that specific marker, not a different exosomal marker?
Am I going to answer?
Yep, go for it.
Okay, so CD63 is very exosomal membrane marker, right?
Yep.
And also others, well, there are known other tetraspanin proteins presented on in exosomes like CD9 or CD81. But in our cell model, HD1080, which have been widely used for cell migration study, express abundant amount of CD63. And for that reporter with pH sensitive, and we need to choose the membranous marker to label the floating tag into extracellular area or outside of the exosomes. So those purpose, I mean, those reasons, CD63 was kind of best fit into our cell model.
Yeah, and if I can just add on to that, you know, we started off by using a non-pH sensitive, you know, the typical GFP CD63 or M-Cherry CD63 reporters that other people were using. And the reason we switched to the pH sensitive is it's so localized inside endosomes that it really blows out your system. It's hard to see the secretion unless you quench that internal fluorescence. So we knew it was a good endosomal MVB marker. Some other tetraspanins like CD9 and CD81, we haven't explored so much, but from work I've seen from other people, they tend to be more localized on the plasma membrane. So they might be better markers of like small plasma membrane derived microvesicles. But it's pretty clear that CD63 is, you know, probably 90% in the endosomes in, you know, just about all cells.
Brilliant, thank you. With that, we are now at the end of our hours allotted slot. So I'd really like to thank Ashley, Alyssa, and Bong Huang for all sort of helping with this discussion. I think we've all learned a lot from this. A complete different, for me, a different way of looking at the exosome secretion, and plenty of food for thought. So thank you to you all for this. And also a big thank you to Ken for letting SNEV join in with this journal club. It's a fantastic opportunity, and I think we've all really, really enjoyed it. So thank you.
Well, thank you, Will. Thanks to Ashley, Alyssa, and Bong Hwan as well, and to all of our participants for joining and for the fruitful discussion. So this will be archived on our YouTube channel. Just look up Extracellular Vesicle Club. And please reach out if you have some topics, some papers that you would like to present, just let me know. So thanks again to SNEV, and maybe we can do this again sometime soon. But until next week, thank you all and take care.
Thank you for discussing our paper.
Yeah, thanks for being here. I really appreciate it.